It is a tremendous pleasure today to welcome a friend and colleague, Dr. Thank God Ebenezer. Dr. Ebenezer has a Bachelor of Science degree in parasitology and entomology from Nnamdi Ezekwe University in Nigeria. This was followed by a Master of Science degree in environmental toxicology and pollution management from the University of Lagos. He has a PhD from the University of Cambridge, which is where we met, because I think I had just left that lab a few years before, but was still very much collaborating when Dr. Ebenezer joined, from Professor Mark Field at in the pathology department. Upon graduating, and Dr. Ebenezer's PhD was on the genome of Euglena gracilis, which is a really important microalgae. After he finished there, he joined the Earlham Institute, the Harity Group. So the Earlham Institute, where he worked on wheat transcriptomics and epigenomics, and has recently moved back to take a programming position at Uniprot back in Cambridge. And I have to say that I'm really impressed, thank God, with not only the research you've been doing, but just how proactive you've been about rallying and organizing the genomics community, both and on two very different sort of topics. Because I'm looking at people in the weight room, and some people are really interested in continuing on with the microalgae and really mobilizing that community, but also taking this other really exciting step for food security and leveraging genomic activities for that, and also just capacity building within the African community as well. So I'm super excited to hear about your talk today. I'm going to make you're already a co-host, so you should be able to get started. And just for participants, please put your questions in the chat, and we'll have lots of time to talk afterwards. So thanks so much for doing this. Thank God.
All right. Thank you, Joel. Thank you for having me today. And my name is Thank God Ebenezer, and I'm a bioinformatician at the European Bioinformatics Institute, and I am with the Universal Protein Resource Team, popularly known as the UniProt team. And today I will be talking to us about BioGenome for food systems in Africa towards a genome-guided non-human biological diversity and improvement. And this talk is mainly on the network building, as Joel mentioned previously. I currently help to support two networks, the one with the Euglena International Network, which we inaugurated last week, mainly focusing on Euglena genomics and bringing the community together, and the digital innovation in Africa for sustainable agri-environment. And the idea, I'll be focusing on this, and which is what this talk is all about. And the idea behind the DAISY network mainly is to see how can we use this to kind of support genomics, food security, particularly in Africa. So just a background to the DAISY initiative. The DAISY initiative mainly is a community network to support genome-guided sustainable agricultural practices in Africa. And the main goal for us is to sequence the genomes of endemic biological species in the agri-environment in Africa, mainly looking at from sequence to diversity to variations and also application. And also I must also mention that this network is pretty new and everything is still evolving. And there are so many new developments in terms of where we want to go with it. So, and I'll also be mentioning the series of things we are doing in order to build the genomic community in the agri-environment in Africa. And it's not just about from sequence to application, but also to improve food systems and well-being in Africa. And there are already a couple of communities doing similar things. For instance, we have the African Orphan Crops Consortium, which may aim to sequence a couple of hundred of orphan crops in Africa, and they're also known as the AOCC. So, but the idea behind this DAISY initiative is to see how will DAISY contribute to the global genomic project and also contribute to the Africa biogenome resource I'm going to also talk about this in the next couple of slides, and also this presentation is will be a very short one, and at the end a bit interactive as well. So, and for us, what are the challenges that we aim to solve? For this DAISY network, we aim to achieve the challenges of addressing the goals of the UN Sustainable Development Goal 02. And what is this goal? The main goal here is to achieve zero hunger on the African continent. And this is the 02. And specifically, we are more interested in how are we going to mainly use this, what we're doing, mainly to address food security issues and nutrition in Africa. You know, how can people have very good nutritional foods and also help them in order to eliminate hunger on the continent. We are also not just focusing on the UN development goals. The idea is to also connect this to the African Union Sustainable Development Goals, mainly the Agenda 2063, that aims to address hunger and food security on the continent. And we aim to do this using genomic resources for both plants and also animals and livestock and biodiversity. And so mainly the idea is to achieve food security and improve nutrition, and also promote sustainable agriculture on the continent. So, and this is the main focus of the DAISY project. And so far, we've been having some communication and some engagement with several other partners on the continent, which I'm going to talk about. But on this slide, basically are the states, an overview of the state of infrastructure with regards to genomic activities in Africa. For instance, we have the History Africa, which is the Human Heritage and Health in Africa. It's a continental program that aims to use genomic resources to also help advance health-related activities on the continent. We have the Acacia, for instance, the Accelerated Crop Initiative mainly, and also aims to build capacity and bioinformatics on the continent. We have the AOCC, which I mentioned previously, the African Orphan Crop Consortium. Then we have the West African Virus Epidemiology for Food Security. We have the Biosciences, Eastern and Central Africa, known as Baker INRI. And we also have the International Institute for Tropical Agriculture, and also the African Union African Bureau for Animal Resources. So again, all of these are initiatives that we've identified. They're already on ground doing a couple of things. So we want to partner with them and also work with them in terms of using the existing resources. For instance, the existing resources that History Africa has built for the continent. But how do we begin to do similar thing in biodiversity and also in food and agriculture and nutrition? How do we begin to generate data and use the information to solve food security and nutritional issues on the continent? So overall, we are aiming for this DAISY network to connect with the Earth BioGenome project. So I'm also going to talk about the Earth BioGenome project, which aims to sequence all life forms on Earth through collaborations and other partnerships. So for us, we aim to build and increase collaborations and capacity within the Earth BioGenome community. And we aim to contribute to the genomic resources in Africa. And we're aiming that the data generated from this will help to inform and drive policy and also socioeconomics on the African continent. And of course, while there are several activities ongoing in Africa in terms of genomic and also within the non-human space, there are so many data that are needed to come out from the continent and also used to support a majority of the sustainable development goals, particularly in the agri-environment in Africa. So this is one way we aim to contribute to this program. So who are our prospective partners? As I mentioned previously, we have identified prospective partnership with the African Union Inter-African Bureau for Animal Resources. So this is the African Union arm that has to do with animal resources. We've identified prospective partnership with the African Academy of Sciences. We've identified prospective partnership with the West African Virus Epidemiology for Food Security, and also with the Earth BioGenome project. And finally, with the Pacific Biosciences in California. So everyone here basically has actually been having a series of meetings over the past several months. And we've been talking, there's been so many engagements that have been ongoing. And eventually this engagement is aimed to lead to the white paper mainly because the idea is for us to then put all of these things, these goals and these ideas into a white paper. So the white paper is currently under development and by colleagues on the continent and also outside the continent. So last month, we had a major meeting, mainly at the conference organized by the Sangha Tree of Life. The Sangha Tree of Life is the UK project or program that aims to support sequencing of species to contribute to the basically to the global idea of sequencing all life forms on Earth. And this conference was organized, which is the BioGenomics 2020 conference, was organized by the Sangha Tree of Life jointly with the Earth BioGenome Project, the Vertebrate Genomes Project, VGP, and also the Global Invertebrate Genomics Alliance. So at that conference, we basically had a workshop on the DAISY initiative for the African continent. And at the workshop, we mainly came up with mainly solidify the ideas behind the white paper on basically having a couple of specialists on the continent and also out of the continent coming together to define a specific goal and what we should aim to define, to basically achieve rather. So we had Professor Nicola Moda, who basically is the PI for the H3A BioNet. And we also have Apollinaire Jinkeng from the Roslin Institute and many other, and also the Dr. Simplis Nwala, who heads the agricultural department at the African Union in Addis Ababa, Ethiopia, and many other speakers at the workshop, mainly to define what we aim to achieve with this initiative. And also, as I mentioned previously, all of this is still evolving and still developing, and we're aiming that by the time the white paper is ready, that would mainly inform the next step in terms of what we aim to do and how we go from there. So far, one of the ways we aim to achieve this is mainly to establish a working group on plant genetic resources in Africa, and not just in plants, but also in farmed animal and non-farmed animal, mainly looking at the general biodiversity. And we aim that it's going to be a continental-wide program, which not only generates data for the African continent, but also contributes to the global initiative to sequence all life forms on Earth, and mainly for the African continent, help to solve food security and nutritional issues. So we also, as I mentioned previously, have partnerships with the African Union, Inter-Africa Bureau, and also the UNEP, and also the African Academy of Sciences. So how do we aim to fund the initiative? One of the things we've been thinking about is how do we leverage on existing efforts on the continent to actually sequence some of these species, for instance, the African Orphan Crops Consortium, and how do we leverage on funding from existing projects that will wish to sequence genomes of species in Africa, for instance, through communities within the Earth BioGenome Project. And the final mechanism would be how do we secure funds to actually run the DAISY initiative through applications to the African Academy of Sciences, the African Union, or NEPAD, mainly, which aims to look at several of the partnerships or initiatives or ideas as this one. So with that, this is basically a very short presentation, and I've kind of given a broad overview of where we are and also where we're going. And so far, we've been working on this network, and it's being coordinated by several colleagues on the African continent and outside Africa. And in Africa, these are colleagues who are basically based in Northern Africa, Eastern Africa, Central Africa, Southern, and also Southern Africa. And in terms of the advisory, the network is currently being advised by Professor Apollinaire Jinkeng at the Center for Tropical Lifestyle, Genetics and Health, and Dr. Catherine Ziumo, and also Rob Davey at the Elam Institute. And with this, I would basically like to thank every one of us for listening, and I will be happy to respond to any question you may have in terms of this network building initiative for DAISY and also where we aim to go with this network. Thank you so much.
Great. Thanks, thank God. We've got lots of time for questions. And I'd actually like to start. So Can you give some examples of the types of organisms or sort of target genomes that you think are important to address in this, with this consortium, that you think might be missed by some of the more global, like the Earth BioGenome Project or the Vertebrate Genomes Project? I mean, what are the African targets that are going to be really important for the consortium?
Yeah, so at the meeting in October, I proposed that it's what if we need to sequence all life on Earth, there is need to mainly focus on communities who are around a particular area, who not basically communities around taxa and also communities around a particular geographical area. And for us in DAISY, the idea is to focus on endemic species in Africa. What are the African species? So I will give you an example. I work on a species called Bostrichus africanus during my master's degree at the University of Lagos. And this species is found around the Gulf of Guinea. But then it's called Bostrichus africanus, and there's another species, that is found outside the African continent. So, but this species is endemic to West Africa, and it's a source of livelihood for the communities in West Africa. But the genome is not yet sequenced. So the idea behind DAISY is to engage that conversation, help to drive it, whereby systems like this that are not yet sequenced because they do not have that, global economic significance, but then they have more of a regional economic significance and community or local significance. These are the organisms we are targeting, indigenous organisms that are basically particular to those communities. And hopefully, if these organisms can be targeted in terms of sequencing, it gets us closer to sequencing all life forms on Earth, and it ensures that the benefits that we get from sequencing genomes around the world is also shared in terms of benefits, between the global south and also the global north. And for instance, using this Bostrichus africanus as an example, that means that if there's any genetic resources that contributes to it, then it will help the communities and also help the information available for it. So that is the main aim of this network, because there are hundreds and hundreds of similar species, and that is what this aims to do in the agri-environment.
Okay, great. Are there any other questions?
I had a couple. Thank you for the presentation. Very exciting project. I guess my first question is about assessing the diversity within a species. So my understanding here is you're going to be building assemblies of lots of different species. Are you going to look within species as well to get a handle on genetic variation that might be important for, say, animal breeding?
Yeah, so the idea is to look at things like that, and particularly, and we aim to use the guidelines. I mean, because the Earth BioGenome Project is also putting together guidelines for some of this, from sample collection to sequencing, and also looking at variations and some annotation, for instance. So yes, the idea is to also see how would this inform breeding, in terms of plants and livestock and generally.
And the other one is just some of the details about assembly. So I'm a bit out of, my knowledge on this subject is a bit out of date, but when I see you embarking on this project now, I can see that you're going to, I think, benefit from a lot of advancements in sequencing technology that have been made. So the assemblies you generate could very well be much better than some of the existing assemblies for cattle, for example. Is the plan to focus on long read type technologies?
Yeah, so the plan is to use long read where we can. For instance, I don't know, you could see the Park Bio, they're already a partner, well, a prospective partner, because they actually have been joining us in the meetings, and they're interested in saying, okay, how do we work together in terms of advancing this on the African continent? So yes, the idea is to leverage on that in terms of long read, because by the time you begin to look into plants and some animals, then you begin to have, there's need to actually have those long read requirement to help with the assembly process.
And my last question is about data sharing. So When could we get access to the outputs of this project? Will the raw data sort of find its way into the typical databases as it's generated, or will there be sort of releases of final assemblies?
Yeah, so I think with these specifics in terms of the data as it begins to emerge, these are things that I think would be discussed, eventually as we begin to finalize the white paper. But I'm hoping that the data availability would basically be in line with what we have with other global genomic projects. For instance, with the VGP, once an organism is sequenced, is released, and I think today they actually sequenced a bird species. So again, with some of this community, because the idea is basically how do you get the data out there and so that it's available and people could actually make use of it. But then ultimately this will be based on what the community within the data community think is the best way, basically. But I would imagine that yes, it's going to go in the line of okay, we sequence it and then we'll make it available so that other scientists could work with it.
Can I ask a question?
Thank you.
Oh, sorry.
You're welcome.
Who's dropped out and has come back in. So maybe more of a community engagement and science sociology type of question. I mean, you're involved in making sort of large scale networks, one focused on Africa and one that's not. Are you finding any differences in terms of working, governance, networking, and so on, and how much of the actual data generation do you imagine happening within Africa in terms of the universities and the capacity building there? Yeah,
so one of the things we did, because the idea is I had a chat with the chair for the EBP and other chairs as well, and also other institute heads. One of the things we are looking at and proposing in this network is that the data generation will happen on the continent. Because first of all, it helps to build the capacity, and also second of all, it helps to ensure data ownership for the scientific communities in Africa. So, and in doing that, we have gone through the African Academy of Sciences, mainly try to look at what is on ground by having distributing a questionnaire and sharing across across several networks within the continent. And we have an idea on what is available in terms of capacity and in terms of sequencing technology. So yes, that is basically going to be the focus, whereby things are done on the continent. Then also in terms of differences between the international network that I'm helping to build and also the African network. So one of the things I see, for instance, with the international network, of course, I already have a couple of, the infrastructure is there already, and several of the network and things are also there. But in terms of building a whole continent-wide program and trying to also push it, so there are also a bit of differences between the two of them, but they're also very complementary. And it ultimately it has to do with engaging with people, speaking with the right set of people, and also making the goals very clear and defining where we think we need to go. And more importantly, trying to make everybody within the network, have ownership of the network, be part of the community. Then eventually people kind of connect to it and, go with it. So that's one of the things, the similarities and also the differences that I see in this.
Are there any other questions from the group? If not, thank you so much. It's a really exciting, initiative, I think, and I'm looking forward to seeing how it's going to develop over the next few years.
Yeah, thank you, Joel. And I must say this is a very short one because everything is still evolving within the network, and we're still at the early stages of building the network. And I'm hoping that yeah, in the future, we'll begin to develop this further and more particularly begin to answer some of the scientific questions that we are laying the groundwork for this initiative.
Great. Thanks so much, everybody. And we'll see you in 2 weeks.
Thank you.
Okay, thanks.
Bye-bye.